Hello, everyone, and thank you so much for joining us. And happy LEMS Awareness Day to everyone. We are so excited to be hosting our first virtual summit and our first presentation with that. We do have some people filtering in right now, so thank you for waiting. And we are getting ready to kick off. We're going to have a presentation with Price Woolridge, our board president and founder. And unfortunately, Dr. Lennon had a scheduling conflict today, but we are very thrilled that Dr. Stephen Meriney is here as well. So, Price, do you want to kick us off with a couple words?
Thanks. And just so everybody knows, Madeline put a lot of work into this. She's got a lot riding on it. So we want to thank her for that. And welcome everybody on behalf of all the board members for the LEMS Family Association. I'm going to recognize them just because I think that's important too. Besides myself, there's Julianne Kostelski in Colorado. She's a founder, secretary, and board member. Lori Dunham is in Florida. She's our treasurer and a board member. Tracy Sharp is in Kentucky, a board member. Devin Lawson, who's kind of overseeing our webinar today, is in Montana and a board member. And then I'd be remiss if I didn't mention Christina Patafio in New York. She was a founder and a board member emeritus. Thanks to all our board members. We have a number of volunteers we don't have time to acknowledge today, but we really appreciate all our volunteers as well. I just want to mention a couple of things real quickly. This is LEMS Awareness Day. This is March the 30th. If you're not aware, and we have a lot of new patients that come on board. So if you're not aware of why we picked this day, it's because in 1957, on March the 30th, Dr. Lambert and Dr. Eaton out of the Mayo Clinic in Rochester published their paper in JAMA, which is the Journal of American Medical Association, where they differentiated their EMG findings for this new disorder yet to be named Lambert-Eaton and myasthenia gravis. It was a landmark day, a landmark article, and that's why we picked March the 30th to celebrate LEMS Awareness Day. All of you are patients. I'm a patient as well. Devin's a patient. So at any rate, a couple of other quick things. We do have a new website. It's lemsfamily.org. It's at the same location, but there's been a lot of developmental work done. It's an entirely new website. We've been tried to include resources that are not duplicated elsewhere. So we would hope that at some point in time, when you have the opportunity, go to our website, lemsfamily.org, and check out the stuff that we got for you. We also gave out 150-ish yellow LEMS Awareness Day pins. So for those of you who requested those and got them, thank you. We did run out of them. But we'll probably do it again next year. So with that, that's all I've got. I'm going to turn it back over to Madeline. And Madeline, thanks.
Great. Thank you so much, Price. And I just wanted to do just a quick little update about the organization since we do have some new people joining us today for our LEMS Awareness Day. All right, so we are a nonprofit patient advocacy organization that's dedicated solely to supporting the LEMS community. We have about 300 LEMS patients and supporters worldwide in our contact database. And we focus on building supportive and educational programs and resources. And we've been doing that for a number of years through our patient chit chats. But then this year, we've also added the LEMS Learn, which we're hoping to host in a total about 4 to 6 quarterly webinars or every other month webinars. We did have one in January that's on our website about navigating physical therapy. This will be going on our website as well. And then we're hosting our virtual summit. So we are in need of volunteers and everything. So if you are interested in being involved more in the Family Association, please send me an email. But we are using a new software today. We've upgraded to Zoom Webinar. So I just wanted to give a little quick background. You can ask questions. The Q&A question is down here. It's on your screen, but I just showed a picture here at the bottom of this slide. You can submit questions at any point during the webinar, and then we will read through them at the end and submit those questions to Dr. Meriney. You can also use reactions if you want to, applaud, show your appreciation or agreement. You can do that as well. Your camera and microphone are turned off. We'll answer questions, like I said, during QA times. And if you're looking for that chance to connect with patients, we will be hosting our next patient chit chat on April 25th. And then just one quick other reminder, we do have this evening another LEMS Awareness Day presentation. And, it's really important for us to focus on research and medical information, but at the same point, addressing the psychosocial issues that also come with the LEMS diagnosis. So we're really excited to have Jody Taub, who's a licensed clinical social worker, joining us. She has a rare disease as well, and she's going to do a presentation on maintaining friendships with chronic disease. So that will be at 8 o'clock tonight. And you can register for that still on our website if you haven't done so already. But with that, I am very proud and excited to introduce Dr. Stephen Meriney, who's been a longtime LEMS researcher. In addition, he is a professor as his day job of neuroscience at the University of Pittsburgh, which isn't too far from me here in Philadelphia, but very important to the organization. He was our first member of our Medical and Scientific Advisory Board. So at this point, I'm going to send it over to Dr. Meriney, and he can start his presentation.
Thank you so much, Madeline. It's really an honor to be invited to speak today. I just want to share my screen so that we can show some slides today. Is this showing up okay on your end, Madeline?
It's still the slide layout.
There we go. How about that?
It's the slide preview, so it's the...
Swap it again. How about that?
Perfect. Great.
Great. Thank you for having me. Today I thought I would talk a little bit about current perspectives as well as emerging therapies. And the reason that I chose those two topics, the current perspectives, we have some more fine-grained information based on some of the research that's gone on in the past 10 years or so on exactly what happens in LEMS and how the neuromuscular system is altered. And then, of course, there have been some new ideas about how we might treat LEMS. And so I want to talk a little bit about that as well. And I'm happy to answer questions as we go, or at the end, I guess, is when we've reserved it. So feel free to drop a question in. Just a little bit about me. I have been at the University of Pittsburgh now as a professor in the Department of Neuroscience since 1993. So 33 years here in Pittsburgh. Before that, I did my postdoctoral training at the University of California at Los Angeles. And there at UCLA, at the time I was there in the late 80s and early 90s, we had a Jerry Lewis Neuromuscular Research Center, which was a building on campus that was devoted to studying neuromuscular function and disease. And I spent 6 years there, which is where I really was introduced to not only the neuromuscular junction, but also Lambert-Eaton syndrome. So it has really been a long time that I've been interested in this topic and this disease and doing research to try to understand a little bit more about what causes LEMS and how it might be best treated. And so therefore, this has been, a process where I started off as a basic scientist trying to understand how the neuromuscular junction works and what happens when it doesn't work well. And so it's been a process that has laid the foundation for then talking about how we can perhaps develop new therapies to try to treat this disorder. Once you understand how the system works, it's easier to break it down into its pieces and decide what you can do to help. And so that's kind of been my strategy over my career. I want to say that a lot of the basic science that I'm going to talk about is based on a mouse model for LEMS that really has significantly contributed to how we study the disease and how we understand some of the ways in which the neuromuscular junction functions in healthy and diseased conditions. And this is really based on donation of blood from patients who have LEMS. We basically purify the serum from the blood and inject that serum, which contains all the autoantibodies from LEMS, into the mouse, and basically passively transfers those antibodies to mice, and the mice then become weak. And so first off, I just want to thank all of the patients who have contributed to these many years of studies by donating blood to this type of work that allows us to generate mouse models of LEMS, because honestly, some of the experiments we can only do in an animal model. We can't do experiments on people. And so the best way to understand what's happening is to create what I consider a very effective model of the disease in a mouse. And this is really the foundation for what we're doing. I want to start with the healthy neuromuscular junction so that I can break down what goes wrong in Lambert-Eaton syndrome and some of the new ideas we have about what's happening that go beyond what I would call the generally understood problems that occur. Of course, we're going to be focusing on the neuromuscular junction, which in this diagram is just shown on the right side, and I might be able to get a laser pointer so I can point to things. So I'm going to blow up this area of the neuromuscular system so that we can look at what's going on. And when you do that in a mouse, and this again is a mouse tissue that shows a neuromuscular junction in a mouse, and we've really stained it with two colors here to emphasize two important elements. One is the red, which is the postsynaptic or the receptors on muscle cells that sense chemical or acetylcholine. And the other is green. And all those green spots are places where acetylcholine or transmitter can be released from the nerve onto the muscle. And one thing that's clear is that there are hundreds of spots that can release this chemical onto the muscle. And so one way that a neuromuscular junction is very strong and reliable in a healthy situation is that there are hundreds of places to release acetylcholine. And we generally don't need all of those to participate every time, because perhaps only about 10% of that capability is required to cause a muscle to contract. And that gives the neuromuscular junction reliability because it's only using 10% of its capability every time. And so you can usually in a healthy situation rely on enough of that chemical to come out, give you a nice muscle contraction. But if we break down one of these green spots even further, some of the new information that we have is occurring within each of these release sites. So each of these green spots, and if we zoom in on one green spot here, circled as an example, and just draw a diagram of what we see, it's easy for me to describe the LEMS situations that we've discovered more recently with this diagram. So each of those green spots really is a really very well organized array of proteins shown as yellow and purple spots here that include the calcium channels that are so important in Lambert-Eaton syndrome. And then, of course, the chemical, the acetylcholine that's dumped out of your nerves onto your muscle comes in these blue spheres. And there are usually two of these with every one of these little green spots. And so a green spot is a highly organized system. And that's important because this ordered structure turns out to be very important for normal function, and that's disrupted in Lambert-Eaton syndrome. The other thing that I want to use is a measure of how strong anyone's nerve muscle contact might be. And so if we are to measure what's happening at one of these contacts between nerve and muscle, we use that with electrophysiology or an electrical way to measure the electrical changes in your muscle cell. And this is what's shown here. So these little lines that look like mountain peaks, this is just the measure of how strong your synapse is, how strong your connection between nerve and muscle. And this is a measure of the way that the muscle might contract. And this triggers that contraction. Now, this usually occurs with 60 or 70 of these blue spheres being dumped onto the muscle. And, with every stimulation, that's only about 10% of the capability of this entire synapse. And that allows for reliability. And so, I want to kind of dive into a little bit about what we know about what happens in Lambert-Eaton syndrome. And one of the reasons that we know more about how to treat it is that each of these purple channels is maybe a calcium channel. And now we know some of the new information is there are not very many of these purple calcium channels in any one release site, any one place that releases your acetylcholine. In fact, the new data suggests that there might only be 3 or 4 of these channels, and they don't open very often when activity occurs in your system. When you try to move your muscle and an electrical spike goes down to the end of your nerve, you might only open a small fraction of these purple spots or channels. And so one reason that the neuromuscular junction is so sensitive to losing some calcium channels is there's just not that many to begin with in every release site. And so even if you lose a few of these, you're going to really compromise that transmitter release site's ability to keep up. And even when one of these channels does open, they don't bring in a lot of calcium. And so you often need more than one of these to open in order to get enough calcium to cause that transmitter release to be occurring at these release sites. And so what we understand now is that there's 2 reasons why neuromuscular junctions are so susceptible to LEMS. One is they don't have very many calcium channels in every release site to begin with. And even when one of those calcium channels opens, it's not very reliable at causing enough transmitter to come out to cause muscle contraction. And so when you have Lambert-Eaton syndrome, you have autoantibodies that are circulating in the blood, and they are attacking those calcium channels. So normally this is a cross-section of a nerve terminal with calcium channels and those transmitter-containing vesicles. And whenever activity occurs in the system, those channels open and bring calcium in. But these antibodies remove these channels and they break them down so that you have fewer calcium channels. And that does not release enough acetylcholine to bring that muscle cell to contract. And so if we look at it back at the level of the single release site that I showed a minute ago, you take this normally well-ordered release site with 4 calcium channels, releasing a lot of transmitter, and then in Lambert-Eaton syndrome, you lose some of those calcium channels. Now your body tries to compensate, and there might be other ones like this red channel. Your body's trying to upregulate to keep up. I think you come to some equilibrium where the disease is taking away channels and your body's adding channels, and you come to some stable equilibrium eventually. But because you have fewer channels, and because the destruction of those channels and other proteins disorders this array, so normally you have this nice 2-row array of proteins, that gets disordered. Both the loss of channels and the disordering of the array reduce the magnitude of the communication that occurs. And so with this drop in magnitude, that's when sometimes muscles can't contract. And so I wanted to focus on kind of the fact that another new piece of information, not only do we have few calcium channels here, but this attack is also going to attack other proteins, not just calcium channels. Of course, it's very common for clinicians to measure calcium channel antibodies. But one thing that you may know is that there are patients that are called seronegative, meaning they don't have calcium channel antibodies, yet they still have Lambert-Eaton syndrome. And so what is now being more and more appreciated is that Lambert-Eaton syndrome is not only antibodies against calcium channels, but it's antibodies against other proteins in this transmitter releasing structure. And so this part of the nerve that's releasing acetylcholine is losing more than just calcium channels. And that could be one, because you actually make antibodies to many other proteins that are here. We just don't measure them when you go get a test at the clinician. And 2, an attack on a calcium channel is also going to have some collateral damage, and some of the neighboring proteins will be affected as well. And so as people have looked at which kinds of antibodies people have and what kind of deficits exist in these releasing structures, I don't want you to be kind of caught up in this maybe overly complicated diagram, but if we break this organizational structure down into single proteins, all the proteins that are labeled here, calcium channels have two subunits. They're both beta and alpha 1. They're both targeted by antibodies. We have proteins called synaptotagmin that are sensing calcium and causing the acetylcholine release. They're attacked by Lambert-Eaton syndrome antibodies in some patients, and as well as some other receptors on the nerve that are also attacked. And so, we have this attack on many different proteins. And furthermore, we have this disorder in the structure here. And so really, LEMS is not just caused by the removal of calcium channels now. We understand that many proteins are attacked. Some patients obviously don't even make antibodies to calcium channels, but still have LEMS. Those are the seronegative patients. And the organization of that releasing structure called an active zone causes weakness as well, because disorganization causes the proteins not to be next to each other the way they were normally. And that small distance change has a big effect on the ability of the synapse or the connection between nerve and muscle to work. And so that's very, very, very important now. And it's clear from some of our animal studies that just removing calcium channels is not enough to even create a mouse that has Lambert-Eaton syndrome symptoms. So we can block calcium channels in a mouse with drugs, and it's not the same as if they have an injection of serum from a LEMS patient. And so it's clear that the mixture of antibodies in the blood of a LEMS patient is important in giving the full description of what causes the disease. That's kind of new perspectives on what we understand. One, that there's very few channels there, and two, that there's this multi-level attack and disruption that occurs. And I now thought I might turn to some of the emerging therapies that people have been using. I'll start off with kind of the current treatment and then move on from there. First of all, of course, if a tumor is present, the focus is on treating the cancer. And in fact, successful treatment of the cancer does tend to reverse the symptoms of LEMS as you remove the trigger that was causing the antibodies to be generated. Of course, the most common therapeutic approach is the symptomatic treatment that is 3,4-diaminopyridine that comes either as Firdapse or Ruzurgi, and these are just different forms of the same active molecule. In the case of Firdapse, this is just a salt form, a phosphate salt. And so, both of these will basically target potassium channels. They block a small fraction of potassium channels, which effectively makes the electrical spike that triggers chemical release from your motor nerve terminal broader. And with this longer lasting spike, you open more calcium channels. So that's because potassium channels are what really brings that spike to an end. And that spike in the nerve comes down and ends quickly because of potassium channels. If you block a small percentage of them, then that spike lasts longer, and that gives calcium channels a greater chance of opening. Normally, calcium channels are actually quite sluggish to respond to such a spike. And so they're very sensitive to the length of that spike. And so instead of having, let's say, an active zone that release site might have one channel opening, if you put on DAP, you'll get many channels opening. And this is one way to boost the calcium that comes in and essentially, recover from that decrease in transmitter release. I see something in the chat, but I don't know if I should wait to the end. But I can address this quickly from Price. Is there another blood test beside voltage-gated calcium channels that could be a marker for LEMS and help with diagnosis? Yeah, that's a good question. Something we've been actually interested in doing. Rather than just test for calcium channel antibodies, we thought it would be useful to develop a panel of antibodies that might create, in a sense, a greater detailed picture for the clinician. But to date, it turns out that because LEMS is not a blood test only diagnosis, it's really a clinical diagnosis for which a blood test provides supporting evidence. The clinician will run through a variety of behavioral analyses, as well as measuring usually the EMG in the hand muscle, doing the so-called repetitive nerve stimulation test. And those things in combination with blood tests provide the diagnosis. I think it would be useful to have what I would call a collection of antibodies that we could look for. But I do fully expect that every patient will have a different mixture of antibodies. And so it might be that there would be some collection that is variable depending on the patient that is more definitive in Lambert-Eaton syndrome diagnosis. But that has not yet been developed. Various people have identified the various other antibodies that can be found, but it's been an interest of mine to conduct a study to try to fully characterize all of the antibodies. And we haven't found that the funding organizations have prioritized that for us yet. So that hasn't been a priority as yet, although I think it's an interesting question. When you have more than one calcium channel opening in those release sites, then that basically takes your normal spike that is very brief. I've just pictured it here at the top. This action potential is the spike in the nerve and made it last longer. And that's what DAP will do, or 3,4-diaminopyridine. And that makes the calcium entry much larger. And this total increase in calcium entry is what gives you this boost in strength. More calcium gives you more transmitter output, and it's a nonlinear relationship. So even small changes in calcium give you large changes in the magnitude of transmitter release. So that's really good. Of course, even though the clinical effects of 3,4-diaminopyridine can be very significant and effectively control symptoms in many patients, it's dose limiting to prevent side effects. And therefore, in studies that have asked patients about their symptoms while taking a form of DAP, most indicate modest effectiveness. And that often leads people to seek additional treatment options. And one of them is mestinon, or it's a molecule called pyridostigmine that really makes the transmitter last longer after it has been released. This is a common drug for myasthenia gravis, but it has hit and miss effects in Lambert-Eaton syndrome. On average, in fact, when studies have been done, controlled clinical trials, it hasn't been found that adding mestinon to DAP gives a very significant effect. So it doesn't look like on average it's usually effective, although individuals might find it effective in the particular cases. So it hasn't been really a game changer in the Lambert-Eaton syndrome field in general. Of course, about 50% of LEMS patients also turn to some form of immunosuppressive therapy, which can also help. Of course, the downside of immunosuppressive therapy currently is it's not selective for Lambert-Eaton syndrome immune system problems. It's not targeting just those antibodies that are attacking the neuromuscular junction. And so you can have side effects, and it can also lead some people to have susceptibility for infection. And of course, in tumor-associated LEMS, impairing the immune system is generally considered not as favorable, because we're trying to rely on the immune system to help fight the cancer as well. And so immunosuppressive therapy is there. It's used very often. It often is helpful. And a combination with DAP can be very helpful for many people. But it's not a silver bullet that will directly target what's going on. So that's kind of what I would give as a summary, but there are some emerging ideas that I think are exciting on the frontier. And one of them is a drug that will help accelerate the breakdown of these pathogenic autoantibodies. And if there's a shorter half-life for the antibodies that are pathogenic, then that might also improve symptoms. And so this drug has been tested in a few case studies, has not been engaged in a Lambert-Eaton syndrome clinical trial as yet, but in the few case studies, it has been effective. It is expensive, and it might only be used in kind of severe cases that might be refractory to the DAP treatments, but it is something that is being developed, and I think shows promise. Another one that's something actually my laboratory is developing is a new symptomatic treatment. And the idea is that DAP targets the potassium channel and it makes the spike in the nerve last longer. And that opens more calcium channels, but those calcium channels don't bring in a lot of calcium. And what we've done is directly target the calcium channels by developing a new compound that holds the calcium channels open longer. I think of this as a foot in the door drug that when the calcium channel opens, normally very briefly, this drug called GV58 will put a foot in the door of the channel and prop the channel open and allow more calcium to come in. And that working with DAP actually can restore transmitter release levels back to normal. I'll show an image of that in a minute. But even working by itself, it's an alternative way to bring in more calcium. So DAP might increase how many channels open, but GV58 will make the channels that open stay open longer and bring in more calcium. And so we're trying to now bring this drug along to clinical trials, hopefully within the next couple of years. And the idea is to have two options, either GV58 and or DAP, or use them together as they appear to be synergistic. And I'll show you that in a second, meaning that when they're used together, there are two different ways in which they work together to give you an even better effect. But before I get to that, I just want to mention there is a new emerging therapy as well that is innovative and selective immune modulation strategy, and it's called CAR-T cell therapy. And this may offer a potential cure for LEMS, although it's very early in development. And the reason this works is that these T cells, called CAR-T cells, can be selectively modified to attack only the parts of your immune system that are making the pathogenic antibodies. That selective targeting requires some gene editing to these cells. And so, that's a complicated process. So far, there are only a couple case studies for Lambert-Eaton syndrome use of the CAR-T cell therapy. There are some risks. Side effects can be significant, and this is also going to be a very expensive therapy. So right now, it's talked about as something for the most severely affected patients. But I think in the long run, this could be an interesting therapy to watch to see how it's working. It's currently being used for other diseases right now, also with success. And so I think CAR-T cell therapy is potential interesting therapy for LEMS in the future. Last thing I wanted to talk a little bit more about this drug that we're developing, GV58. If you recall, I showed an image of the activity in muscle cells when the nerve is active. And this activity is this large mountain peak. In a healthy neuromuscular junction, that mountain peak is very large. This is an electrical recording from muscle cells. If you inject those healthy mice with serum from Lambert-Eaton syndrome patients, then this large amount of electrical activity in the muscle drops significantly. And this is really what happens in LEMS, that the normally strong depolarization or the strong electrical signal in muscle drops so that the muscle doesn't fire an action potential and contract anymore. If we add GV58 alone to that, we can double the size of that transmitter release. And that's the same as different forms of DAP. They basically double how much transmitter release is coming out of your terminal. Either of these can do that. But interestingly, when you use them both together, they're synergistic. And added together, they can completely reverse the deficit in transmitter release. And so I think the strategy right now is to test the safety of this approach. And right now we're in an animal testing phase, and we're doing some of the effectiveness testing and the safety testing, and we're seeking funding to try to bring this option to patients. I've spoken with Catalyst many times about this. I think it's the next generation, but I have to, in order for us to work with Catalyst on bringing this to patients, we have to get past our own internal testing for safety and effectiveness, because pharmaceuticals don't usually want to invest in anything until it's ready for the clinic. So we're currently seeking funding for that and applying to federal agencies for support. That really is my summary. I wanted to leave a lot of time for discussion and questions. I also have also posted my email address here. Happy to receive later. If you think of something later, you can send me an email. But maybe what I'll do is stop now and then let Madeline tell me if there are any questions.
There are.
Okay.
And just a quick thank you so much, Dr. Meriney. I speak on behalf of our patients. I'm sure they're all thinking what a rewarding experience it is to hear from someone who has dedicated their career, working in that neuromuscular junction area, specifically LEMS. So I'm sure this was a very exciting presentation for them. And it also brings hope for patients too, as you're talking about the the emerging therapies as well. So a big thank you for your time. But we do have some questions, and I'm actually going to kind of pose the first question to you. And as a patient advocacy group, or as a group of patients, what can we do to help bring these emerging therapies from, studies or being at the lab bench, out? Is there something that patients can help support? How can they share their story? What is beneficial for you in this process?
I think there's really two ways patients can help. The first is in order for us to convince the federal funding agencies that we need to develop new therapies for Lambert-Eaton syndrome, we're developing a patient survey that will essentially allow us to ask, how happy are you with the current therapies that you are using, and do you think there's a need for new and improved therapies? And so one thing we're doing is developing a survey just to ask patients how needed is something new. And we have to demonstrate need in order for the federal agencies to believe that it's worth investing in. So that's one. And I'll prepare that questionnaire once I get all of the approvals through the IRB, et cetera. I can start making that available for people who might be interested in participating in that.
Wonderful.
Second would be and this has to be done through your clinician. If you are undergoing plasmapheresis, which most LEMS patients do not do in the United States, but if your clinician and you have both decided that that's something you are going to do, I would ask you to ask your clinician to store the serum that's collected during plasmapheresis in the freezer, and then to contact me so that I can establish an IRB protocol with that clinician to use that serum in the animal testing. We need serum in order to generate a Lambert-Eaton syndrome model mouse, and it takes quite a bit of serum to do that. If you're not having plasmapheresis, it's usually not easy for you to donate enough blood to matter. So it's usually just the small blood. The type of blood that's collected for typical routine blood testing is not very much, and that doesn't usually make it worthwhile for establishing an IRB protocol to collect that blood. So I would say only if you're undergoing plasmapheresis will it matter.
All right. Perfect. Thank you very much. And that kind of addresses one of our first questions about if patients can donate blood for your study. So thanks for checking that one off our list.
No, I could qualify that by saying if your clinician's very interested in collaborating with us, we do still take blood, even though it's not plasmapheresis, from a few clinicians who are interested in collecting blood over time, because it takes a while to develop enough. So those people, I would have to work through the clinician to establish this protocol.
Great. Perfect. And I think this is a very important question that's also on a lot of LEMS patients' minds. But the antibody that is causing LEMS, is there a specific reason why it's being produced, or is that kind of the million-dollar question that we're looking for an answer to?
That's a good question. And of course, we know for the tumor-associated LEMS that the tumor that is commonly associated with LEMS is a small cell lung cancer. That cancer is a neuroendocrine cell that has become malignant. And those cells that are lung cancer cells for small cell lung cancer have the same calcium channels. And so when the body is attacking the tumor, which it will do, it will recognize that the cancer is a cancerous cell and it should be attacked. It's making antibodies to attack the tumor. Those just happen to cross-react with the neuromuscular junction. For non-tumor associated LEMS, we don't know why calcium channel antibodies are made or other antibodies are made, but it's very often the case that in non-tumor associated LEMS, those people sometimes are susceptible to autoimmune diseases and may have other autoimmune diseases as well. And so for some reason, that's, as you said, unknown, their own immune system is not recognizing the calcium channels as self. It's thinking it's foreign, and it's making an antibody against it. But we don't know in that case.
Great. Our next question is kind of asking about the calcium channels and any other proteins. Are these permanently damaged by the LEMS-related autobodies or antibodies? I'm sorry, not autobody. And does some type of recovery happen?
Yeah, so it's believed that they are lost, but that the motor neuron that is constantly making more. First of all, calcium channels don't last forever in the membrane anyway, even in the healthy condition. They turn over on a regular basis. And so your motor neuron is always making new calcium channels, and those that you have are occasionally turning over, maybe with a half-life of days or weeks, but they're going to turn over eventually. And so with the Lambert-Eaton syndrome antibodies maybe damaging them or removing them from the membrane, the cell is constantly trying to replace them. And so, yes, they're lost, but they're replaced. And then the reason that you don't fully, and that's why a tumor-associated patient recovers when the tumor's gone, the antibodies disappear, and the motor neuron just makes more calcium channels, and you get back to a healthy state. In the Lambert-Eaton syndrome case where you're a non-tumor patient and the disease continues, it's just you reach a balance between the antibodies taking them away and the nerve making more. And that's where you come to some equilibrium.
And does this variation in antibodies, does that cause the severity of the disease, in your opinion?
So we've measured what the concentration of antibodies people have to calcium channels across different patients, and it's quite variable. Some patients have a very large number of antibodies to calcium channels. Some patients have very few. But what we found was the level of calcium channel antibody, the specific amount of calcium channel antibody you have, does not correlate with the ability to transfer that disease to mice or with the severity of your symptoms in the clinic. It's just the presence or absence. And we think that's because it's not just the calcium channel antibodies that are causing the trouble. It's the mixture of antibodies and the consequences of those specific antibodies you have, how much they're damaging your nerve terminal at the end of the muscle connection. So it's the absolute levels of calcium channel antibodies we found are not 100% predictive for clinical problems.
And kind of a similar question, I think, but is there a reason why LEMS affects some muscles more than others, especially in more severe cases?
That's a great question. And honestly, that's one of the questions that has tormented us for decades. We have no really good answer as to why, not just LEMS, but many other neuromuscular diseases, why they selectively target certain muscles and not others. And you may know that the same exists in myasthenia gravis, where there might be different muscles that are targeted. Eyelids are targeted in myasthenia gravis, eyelid muscles, but not other muscles. And in LEMS, it's certainly also true that certain muscle groups are targeted more than others. And it's not clear why. And I have not seen any studies that have given a good answer as to why yet. We can propose potential, hypotheses as to what we think is happening. One of the potential hypotheses is that every neuromuscular connection has what's called a safety factor, which means what's the amount of acetylcholine you're releasing normally? Every neuromuscular connection in your body is normally releasing more than you need to get a muscle contraction. And the more that you need is your safety factor. How much safety do you have? How much extra acetylcholine do you release? And that safety factor is not the same in every muscle. And maybe if you have a smaller safety factor muscle, it's more susceptible to LEMS. But that hasn't been proven, and that's just a proposal at this point.
And then one other question that just popped up. I'm sorry, the Q&A is
That's all good.
very lively. But with that exercise, a lot of patients, especially in the last webinar that we were talking about, they get that categorically, like that spike after after working out.
Yep.
And do you have a reason on why you think that happens?
Oh, yeah, that's well understood. So that's called short-term plasticity or facilitation upon exercise. And what that means is that, let's say you normally, if you have LEMS, you have less calcium coming in than you need to cause the chemical to come out of the nerve terminal. Well, if you repeatedly stimulate that nerve terminal very fast, and when you're exercising, every time I hold a contraction, my spinal cord is sending signals at high frequency to the end of the nerve. And every time a signal gets there, a little bit of calcium comes in. But if those signals come right after each other, that calcium can accumulate. And so it's called residual calcium or leftover calcium from the last stimulus, piling on top of new calcium from the next stimulus. And that you effectively are just boosting the calcium in your nerve terminal by repeatedly activating it. Now, it won't last long because the calcium will be handled and gotten rid of from the cell. But for a short period of time, you can recover strength because you have accumulated calcium with repeated activation. And that's a form of short-term plasticity that your nervous system always engages in, which is that during repeated activation, there's always this accumulation of calcium.
Wonderful. Thank you very much for that explanation. Just one more kind of just basic LEMS. Do you see LEMS as being progressive in your opinion?
So
in the case studies and stuff that you're reading?
Yeah, so I think there is a progression from first diagnosis until a stable state later. I think when I think of progression, I think of other diseases that continue to progress to complete failure. LEMS usually doesn't do that. LEMS will progress from initial diagnosis until a more stable state later. But once that has been reached, as I said, I think you get this equilibrium between what the nerve is doing to try to compensate for the attack and what the antibodies are doing to attack. And eventually that comes to a balance. And so LEMS patients don't get to the point where transmitter release goes away completely and just progresses to complete failure. It progresses to a point where I think is a balance between what the nervous system can do to help and what the antibodies are doing to take it away.
Wonderful. And we did get one question about LEMS current treatments, and that's IVIG. And I know you talked about immune suppressants. Would you put IVIG into that category as well?
So that's an immune modifier. It's not a suppressant. And I think it's very effective for people because it can provide some benefit. I think that, let me just see if I can give a better answer. In general, it tends to reduce your immune inflammation, and that can help, make the immune attack less severe. So you're giving extra immunoglobulin. So IVIG is you're giving more antibodies, but they're not pathogenic antibodies. So you're basically giving a bunch of antibodies that maybe overwhelm the pathogenic antibodies to some degree and reduces the inflammation, and that can give you relief. I don't think I would have predicted that, but it works.
And I'm sorry to just take you back to one other question that we just had to come in, and then we're going to have one or two questions. We'll conclude this section of our webinar. But one question is asking about that crash that sometimes patients get after overdoing it? And is that because of, what you've mentioned before, or is that a different reason that patients are
Overdoing it, you mean physically? So after
yes.
if you overdo it physically, two things happen. In the short term, you can have this boost of calcium, which makes you stronger. But of course, every time you overdo it, you're also releasing a lot of chemical, and you can have some short-term depression in your function, which could, be due to depleting some of your ability to release. So after exercise crash is usually like a fatigue of the neuromuscular system.
Mhm. And then one just final question. You mentioned the case study that's going on about efgartigimod, or what's known
Yeah,
as the brand name Vyvgart.
yeah.
Do you know where patients could get more information on this case study? Um,
I can
there's lots
certainly.
of chatter, I know, in the chat, but also we've seen on social media and stuff as well.
Sure. So my sources come from the scientific literature. Um, and I usually use PubMed as my search
Okay.
engine. Um, and what I usually do is I search PubMed, which I think is publicly available, right? Yeah.
Yep. Hmm.
I search PubMed for the keywords like the name of the drug and LEMS, and I look for studies there. If I do a quick search, you'll get 100 or so studies that talk about both things to find the few that are the actual study you want. I'm happy to, look up those studies and send them to you, Madeline, if you want to
Yep.
distribute them to people.
Yeah, that would be great. And just to clarify, this question is asking about a trial of Vyvgart, and there is currently no trial for Vyvgart
No, there's
and
no trial
LEMS.
open, right? There's been case studies published.
Perfect. And can you just tell us the difference for patients between a case study
Oh,
and a
sure.
clinical trial?
Sure. Clinical trial would be one in which a group of clinicians or centers might decide they're going to work together to maybe evaluate tens of patients or 20, 100 patients. And they will divide those patients into two groups, one that will receive the drug and one that will receive just water as a vehicle, like no drug treatment, but otherwise they'll get an injection of something or a pill of something. And in that case, you can compare the populations of patients, one that received the drug and one that got a placebo, and ask whether the placebo and the drug treated are statistically significantly different from one another. Did it make a difference in a population of people? That would be the clinical trial. The case studies, very often what happens is a clinician receives permission to try a drug on one patient. And they will try the drug on one patient, and if they see significant effects, they'll write a paper about it. And they'll say, oh, I gave this drug to one patient and look at what happened. You have to be careful when you read those because you don't know. That's only one patient. And that's a case study. That's a case where they just said, I'm going to try this on one patient. And for some of these drugs, they've tried it on one patient for, maybe two or three clinicians have tried it on one patient, and it worked on all of them. And everybody getting excited, like, hey, it worked on these three patients. But is it going to work on average on over 100 patients when you compare it to a placebo? That's the real question, and that's what people really want to eventually move toward. But I can certainly send you in case of the emerging therapies, it's not clinical trials, it's just case studies.
Great. Thank you very much for that important distinction between the two. And then our last kind of question is, we've actually gotten about your research, and you had mentioned the survey. Do you have a timeline? I know patients are really eager to share their story and share their opinion of when we might be able to see that survey.
Yeah, I was hoping it would be out by now, but as busy as we've been, I need to run this through my patient review board
Hmm.
and develop and get them to approve all of the paperwork associated with distributing a survey to patients. And that has gone slower than I want. My hope is that happens within the next month or so.
Great.
So that's the plan.
And just Dr. Meriney and I have been in conversation that as long as the IRB is okay with this, that we would send it out to patients as well through the LEMS Family Association.
Thank you for that. I appreciate it.
Yeah. Well, and we also got some questions, and I just wanted to tell everyone that this webinar will be posted online. We will email you all when it's available, but there has been lots of comments, Dr. Meriney, about wanting to share your presentation with their clinicians and with their neurologists. So thank you very,
Absolutely.
very much. One other question I wanted to address is that we are working on trying to assemble a list of patients who asking patients who their neurologists are, so we can kind of identify some of these neurologists that are seeing a larger percentage of LEMS patients.
Yes, that would
Um,
be helpful to know which neurologists are seeing a lot of LEMS patients for sure.
yes, so we are working on that, but I just wanted to thank you so much, Dr. Meriney, for spending LEMS Awareness Day with us. I know
Absolutely.
I speak on behalf of the patients that it was just such a wonderful presentation. Thank you everyone so much for joining us. And just a reminder that we do have a presentation today this evening at 8 p.m. Eastern Time with Jody Taub, who's a licensed clinical social worker. And she's going to be doing a presentation on maintaining friendships while living with a rare disease. So thank you so much, Dr. Meriney. Price, do you have any closing words?
Oh, thank you, Dr. Meriney. That was extremely interesting and enlightening, and it was exciting to me. So
Thank you. I'm
really,
happy.
really appreciate your time, and we appreciate everybody for joining us today. It was a large group. We're very, very appreciative. Thank you.
Yep.
Thank you. We really enjoy what we do. And so it means a lot that it matters to people.
Thank
And
you.
Madeline, I just put the link to you in
Yes.
the chat so you can distribute that.
Perfect. Thank you very much. And hope everyone has a great rest of the afternoon, depending on where you are. But we hope to see you for our later presentation. And thank you so much, Dr. Meriney.
Thank you.
All right. Bye-bye, everyone.
Bye.